Many applications of this knowledge are in medicine and biotechnology. Modern antibiotics have been developed based on an understanding of the mechanisms of replication, transcription, and translation. One of the most exciting fields in science today is biotechnology. The developments in this area help us in our efforts to maintain our health and diagnose and combat diseases. Some drugs and vaccines, which were once difficult to obtain, are now synthesized via recombinant DNA techniques. It is hoped that the genetic engineering of plants and animals will result in production of improved varieties of crops and livestock. Who could have imagined all these when Watson and Crick proposed the double helix structure in 1953? Science works that way. Discoveries are made, and many years later, new applications are found for them. Without science, there is no technology. We'll be back to sum up our discussion today when we return from this short break. When we see that children resemble their parents, we take it all in stride and say that it is a matter of heredity. If we had to pin this down to a single molecule, then we'd say DNA, or deoxyribonucleic acid. Genetic information is stored in DNA. This is relatively new knowledge. Nucleic acids had been known for a long time, but no one knew exactly what they were for. In 1944, Oswald Avery determined that DNA was a substance of heredity. This was an extremely important discovery, although not everyone may have realized it then. Why is DNA so important? Our DNA molecules contain all instructions for all the proteins, enzymes, and ultimately, all the carbohydrates, lipids, and other nucleic acids our bodies will make. So, as we've been doing in our study of biopolymers, let's first find out what nucleic acids are made of. Hello, and welcome to another episode of Chemistry in Action. In our last lesson, we studied the biopolymers found in food, carbohydrates, lipids, and proteins. We learned that these are digested in our bodies and converted into other molecules that are used for growth and development and as fuel for our energy needs. Another type of biomolecules, the nucleic acids, are found in our food but are not required for our nutrition. However, they're found in all living things and are essential to all life. Today, we will find out why this is so. We will begin by examining the structure of the nucleic acids, DNA and RNA. We will see why they are called the molecules of heredity. We've got a lot in store for you today, so stay tuned while we pause for some reminders. Two types of nucleic acids are found in cells: ribonucleic acid, RNA, and deoxyribonucleic acid, DNA. Nucleic acids are polynucleotides. That is, they are polymers of nucleotides. A nucleotide has three distinct groups: a base, a sugar, and a phosphate. Let us consider these groups one at a time. First, the bases. The bases of DNA and RNA are nitrogen-containing aromatic compounds. Four types of bases are found in RNA: the purines, adenine, and guanine, which have a two-ring structure, and the pyrimidines, uracil and cytosine, which consist of a single ring. DNA contains the same bases: adenine, guanine, and cytosine. Instead of uracil, however, it has the base thymine. A closer look at thymine shows a methyl group at carbon 5. Otherwise, all other parts of the thymine molecule are identical with uracil. The sugar of RNA is ribose. Ribose is a 5-carbon sugar. Like glucose, it can be written as a cyclic compound. Note especially in this structure the hydroxyl groups at carbon 2 and carbon 3. DNA has a slightly different sugar, deoxyribose. It is almost identical with the ribose, except that it has no hydroxyl group at carbon 2. Because of this difference in sugars, DNA containing deoxyribose is called deoxyribonucleic acid. RNA containing ribose is called ribonucleic acid. Finally, the phosphate group. The phosphate group consists of one phosphorus atom surrounded by 4 oxygens. It was known that DNA was made up of bases, sugars, and phosphates. But how are these parts arranged in the polymeric structure of DNA? This remained a puzzle for many years. In 1953, James Watson and Francis Crick, using X-ray fiber diffraction data of Maurice Wilkins and Rosalind Franklin, proposed a structure for DNA. It was quickly accepted as a correct structure and is popularly known as a DNA double helix, or sometimes as the Watson and Crick double helix. How is genetic information copied and transferred? Let us imagine the simplest case possible. The daughter cell must have exactly the same genetic information as the parent cell. For this to happen, whatever DNA is present in the parent cell must somehow be given or transferred to the daughter cell. To make an analogy, the daughter cell must receive what amounts to a Xerox copy of the parent cell's DNA. This is what happens when bacterial cells divide. But it is not as simple as Xeroxing the DNA. Let us look at the process in just a bit more detail. Just before the cell divides, the DNA double strand separates into single strands. After the double strand has separated, each single strand of DNA serves as a master or template for the copying or replication of DNA. The second strand is built up or synthesized based on this. Nucleotides are added, one at a time, following the sequence of bases found in the parent strand. The first base put in is adenine, and then the next, this time a thymine. The two are linked through a sugar-phosphate bond. A third monomer, a guanine, is brought in, and the cycle begins again. The polynucleotide chain extends this way: cytosine, adenine, and adenine. This is repeated until the entire DNA molecule has been copied. The same process occurs with the other parent strand. Thus, after both strands have been replicated, the product DNA molecules are identical. The entire process is called the replication of DNA. Following the master plan or sequence contained in each parent strand, a complementary strand is built, one base at a time. Two DNA molecules, exactly the same, are the result. There is a point when the parent cell has two copies of the DNA molecule, but very soon after, the cell divides and each daughter cell receives one copy of DNA. We now know how DNA is copied, but does that explain the statement we made earlier that ultimately the DNA molecules contain instructions for all the proteins, enzymes, carbohydrates, lipids, and other nucleic acids in the cell? Let's now try to see how this is so. There is a portion of our DNA that gives instructions for the synthesis of each protein in our body. The DNA that gives instructions for making this protein is a gene of that protein. For example, all of us have the protein hemoglobin in our blood. When our body makes this protein, amino acids don't just link up randomly. The gene for hemoglobin directs the synthesis of that protein. Every molecule of the alpha hemoglobin has a chain of 141 amino acids. Looking at a portion of this sequence, the first 7 amino acids in the F chain of hemoglobin are leucine, serine, alanine, leucine, serine, aspartic acid. Each amino acid is there in a definite place and order because the DNA sequence of the hemoglobin gene specifies it. How can DNA sequence direct the synthesis of hemoglobin? This takes place in 2 steps. First, the information in DNA is transferred into another form. DNA sequence is transcribed into RNA. This step is called transcription. In the second step, the information contained in RNA is processed or translated into protein sequence. This step is called translation. In the past half hour, we've been trying to understand why DNA and RNA are called the molecules of heredity. First, we learned about the structure of the DNA double helix, with alternating sugar and phosphate portions and bases in the middle held together by hydrogen bonds. This model is a basis for explaining how DNA is copied through the process of replication. Furthermore, it is a basis for explaining how proteins are made. In transcription, DNA information is encoded into RNA. In translation, RNA information is deciphered and the protein chain is built. We also saw how this fundamental scientific information gave rise to many applications in medicine and biotechnology. And there, our story on DNA and RNA ends. Next time, we will begin a new series of lessons on the environment. We will start with the atmosphere, the air around us. Until then, this is your host, Ramon Miranda, saying good day. In transcription, the information in the DNA molecule is transferred or transcribed to an RNA molecule. RNA, like DNA, is a polymer of alternating sugar and phosphate groups. The bases adenine, cytosine, guanine, and uracil are attached to sugar, ribose. While DNA is usually double-stranded, RNA is frequently found as a single-stranded molecule. RNA bases can also form stable hydrogen bonds with DNA. The main difference is the base uracil in RNA, which forms hydrogen bonds with adenine. There is no thymine in RNA. Other DNA-RNA base pairs are thymine-adenine, cytosine-guanine, and guanine-cytosine. How does transcription take place? Remember that in RNA, uracil is used instead of thymine. We can represent this by using a different shade of green for uracil. One strand of DNA serves as a template for the synthesis of RNA. The RNA strand is built up one nucleotide at a time, very similar to the replication of DNA. The same cycle of adding and linking nucleotides is repeated until the entire RNA molecule has been built. The newly made RNA molecule then travels to the protein factory sites, the ribosomes. Here, proteins are made. The RNA travels, so it is called messenger RNA or mRNA. At the ribosomes, the message or code, which is the RNA sequence, is deciphered. Nature has a very clever way of deciphering the information in mRNA. The process is called translation. First, an analogy for us to better appreciate this code, as we call it. We know the correspondence between Roman and Arabic numerals. The symbol I in Romans is 1 in Arabic. II is 2, IV is 4, V is 5, VII is 7, and so on. Okay, so think that the numerals on the left column are a code for the numerals on the right. The genetic code relates RNA sequence to amino acid sequence. In this code, 3 RNA bases code for a specific amino acid. Each 3-base code is called a codon. For example, CUG is a codon for leucine. UCA codes for serine. GCU codes for alanine. GAU codes for aspartic acid. Sometimes, several sequences code for the same amino acid. The 4 codons, UCA, UCC, UCG, and UCU, all code for serine. There are 64 possible combinations of the bases A, C, G, and U. 61 of these code for the 20 amino acids. 3 combinations are the signals that stop the protein chain from growing further. Going back to the amino acid sequence of hemoglobin, the RNA coding for the first 6 amino acids would be CUG, UCA, GCU, CUG, UCA, and GAU. We now know about the relation between RNA bases and amino acid sequence in a protein. How is a protein actually made? At the ribosomes, the mRNA recognizes another type of RNA molecule, transfer RNA, or tRNA. tRNA has 3 bases near the middle, called the anticodon, that are complementary to the codon on mRNA. Each tRNA molecule is attached to a specific amino acid. mRNA with a CUG codon needs a tRNA with leucine. The anticodon contains the bases GAC. When these 2 molecules meet, codon and anticodon recognize each other and form stable hydrogen bonds. These hydrogen bonds hold the tRNA molecule at the ribosome site. Then, another codon is deciphered. While the two amino acids are beside each other, an enzyme makes a peptide bond and connects the two amino acids. Now the cell is ready for the next round of codon, anticodon recognition and peptide bond formation. In this way, the entire mRNA sequence is translated until all amino acids are in place. When the last amino acid has been added to the protein chain, the tRNA is released. The protein is now complete. Proteins and enzymes are synthesized this way. Enzymes, as you remember, catalyze reactions that synthesize carbohydrates, lipids, as well as various smaller organic molecules. This is why we say that our DNA molecules contain all instructions for all the proteins, enzymes, and ultimately all the other carbohydrates, lipids, and nucleic acids that our bodies will make. When all the cellular components are thus made, the cell and the organism as a whole functions properly. This is what we know about the mechanisms that transfer information from DNA to RNA and translate that information from RNA into proteins. Is it any use to us?